In the selective permeability activity using starch solution and iodine, students explore how different molecules can move across a semi-permeable membrane. This experiment not only reinforces the concept of selective permeability, but also gives students a hands-on way to see diffusion and molecular movement in action. Diffusion of iodine. Iodine, a small molecule, can diffuse through the semi-permeable membrane, plastic bag. When iodine molecules come into contact with starch, they form a blue-black complex, indicating the presence of starch. This color change provides a clear visual cue to students, showing that iodine has successfully moved into the bag and reacted with the starch. Starch molecules and membrane selectivity. Starch molecules, being relatively large, cannot pass through the plastic bag. This demonstrates selective permeability, where the membrane allows smaller molecules like iodine to pass, but restricts larger ones like starch. Students can see that although the iodine moved into the bag, the starch remains confined within it, visually reinforcing the idea that the cell membrane selectively permits certain molecules based on size. Molecular movement and concentration gradient. The movement of iodine into the bag also illustrates diffusion movement from an area of higher concentration, the beaker solution, to an area of lower concentration, inside the bag. Students witness how molecules naturally diffuse across a membrane to reach equilibrium, a core concept in passive transport mechanisms. Modeling the cell membrane. The plastic bag in this experiment mimics the semi-permeable nature of the cell membrane, helping students visualize how real cell membranes control the passage of substances. In biological membranes, selective permeability is controlled by the phospholipid bilayer and proteins, which allow only specific molecules, based on size, polarity, or other factors, to cross. Importance of selective permeability in cells. Selective permeability is essential for maintaining homeostasis in cells. For instance, cells allow nutrients and ions to enter while keeping out harmful substances. Students can connect this experiment to real-life cellular processes, like how cells absorb nutrients and regulate water balance, or how kidney cells filter substances in the body, allowing essential molecules to pass while restricting others. Why did iodine pass through the membrane while starch did not? This question leads students to think about molecular size and structure. Starch molecules are too large to pass through the pores in the plastic bag, whereas iodine is small enough to diffuse through. What would happen if the concentration of iodine outside the bag decreased over time? This could prompt a discussion on equilibrium, where iodine would continue diffusing into the starch solution until its concentration is balanced inside and outside the bag. How does this experiment illustrate passive transport? Passive transport, such as diffusion, requires no energy. Students can discuss how this aligns with cellular processes where molecules naturally move from areas of high to low concentration without energy input from the cell. Membrane permeability and drug delivery. This experiment can be related to pharmacology, where drugs need to cross cell membranes to be effective. Understanding selective permeability helps in designing drugs that can effectively reach their target cells. Water regulation and osmosis in cells. The activity demonstrates the principles that cells use in regulating water and solute levels, crucial for osmoregulation. In the human body, this regulation is critical for cells to function properly, particularly in organs like the kidneys, which filter blood based on selective permeability. Food and nutrition. The iodine-starch reaction is similar to how digestive enzymes break down starches into absorbable sugars, which can then pass through the intestines. This provides a relatable example of how molecular size impacts nutrient absorption. Conclusion: this selective permeability activity allows students to engage with and understand core biological principles in a tangible way. By seeing how iodine diffuses and reacts with starch inside the plastic bag, they gain a clear visual understanding of how selective permeability works. The activity connects molecular movement, cellular function, and real-world applications, making it a memorable demonstration of how cells maintain control over their internal environment.